Assalamu alaikum. In this lesson, we will do the MCQs of Chapter 2, Biological Molecules of First Year Biology. All material things are made up of elements. All material things are made up of chemical elements. The elements are made up of atoms. The chemical elements are made up of atoms. The bodies of living organisms are made up of some of these elements. The bodies of living organisms are made up of some of these chemical elements. Percent of human body by weight consists of carbon, hydrogen and oxygen. 95% of human body by weight consists of carbon, hydrogen and oxygen. Is a branch of biology that deals with biochemical basis of life. Biochemistry is a branch of biology that deals with biochemical basis of life. In a compound, the bonding of other atoms with carbon atom is called dash compounds. In a compound, the bonding of other atoms with carbon atom is called organic compounds. The molecules play important role in the structure and function of cell organelles. The organic molecules play important role in the structure and function of cell organelles. The skeleton or backbone of large organic molecules is made up of dash bonds. The skeleton or backbone of large organic molecules is made up of carbon-carbon bonds. Both organic and inorganic compounds are present in protoplasm. The most abundant compound found in the protoplasm is water. The most abundant compound found in the cell is protein. Carbohydrates, lipids, nucleic acid and proteins are dash compounds. Carbohydrates, lipids, nucleic acid and proteins are organic compounds. Dash have structural functions in a cell. Proteins have structural functions in a cell. Dash are proteins in nature. Enzymes are proteins in nature. A cell has percent of water. A cell has 70% of water. Chemically, a mammalian cell has dash percent of inorganic ions. Chemically, a mammalian cell has 1% of inorganic ions. There are dash types of fundamental biological molecules in a protoplasm. There are 4 types of fundamental biological molecules in a protoplasm. Water is an compound. Water is an inorganic compound. Dash is one of the best solvent. Water is one of the best solvent. The organic molecules in living things have dash functional groups. The organic molecules in living things have characteristic functional groups. Protoplasm is unable to perform normal functions without dash. Protoplasm is unable to perform normal functions without water. Large organic molecules are called macromolecules or dash. Large organic molecules are called macromolecules or polymers. The subunits of polymers are called monomers. The process of removal of water molecule from large organic molecule is called condensation. Through condensation, the amino acids join to form a molecule called dipeptide. A dash is an organic compound derived from amino acids. A dipeptide is an organic compound derived from amino acids. The monomers of proteins are amino acids. The monomers of nucleic acid are nucleotides. Dash is the reverse of condensation. Hydrolysis is the reverse of condensation. The chemical breakdown of polymers into monomers due to reaction with water is called hydrolysis. Dash reactions need specific enzymes. Both condensation and hydrolysis are reactions which need specific enzymes. Protoplasm cannot survive if its water is reduced to -%. Protoplasm cannot survive if its water is reduced to 10%. Water is dash molecule. Water is polar molecule. The oxygen of water molecule is electronegative. The hydrogen of water molecule is electropositive. Water molecules are bonded to one another through dash bonds. Water molecules are bonded to one another through hydrogen bonds. Hydrogen bonds are dash than covalent bonds. Hydrogen bonds are weaker than covalent bonds. Dash is the universal solvent. Water is the universal solvent. Dash provides medium for most chemical reactions in a cell. Water provides medium for most chemical reactions in a cell. The cohesive force of water is due to its dash bonding. The cohesive force of water is due to its hydrogen bonding. Water molecules dash to surface. Water molecules adhere to surface. Water flows freely without breaking apart due to its dash forces. Water flows freely without breaking apart due to its cohesive forces. Water has dash forces. Water has both cohesive and adhesive forces. Water has dash specific heat. Water has high specific heat. The amount of heat required to raise the temperature of 1 gram of water is called. The amount of heat required to raise the temperature of 1 gram of water is called specific heat. Water absorbs or releases large quantities of heat energy with dash change in temperature. Water absorbs or releases large quantities of heat energy with little change in the temperature. The temperature of water rises and falls dash other liquids. The temperature of water rises and falls slowly than other liquids. The high dash of water helps the organisms to maintain their body temperature. The high specific heat of water helps the organisms to maintain their body temperature. The process of converting liquid into vapors is called dash. The process of converting liquid into vapors is called vaporization. Dash helps animals and plants to get rid of excess body heat through sweating and transpiration. Vaporization helps animals and plants to get rid of excess body heat through sweating and transpiration. Dash bonds in water causes it to remain liquid rather than change to ice or steam. Hydrogen bonds in water causes it to remain liquid rather than change to ice or steam. Without dash bonds, water would boil at -80 degree and would freeze at -100 degree. Without hydrogen bonds, water would boil at -80 degrees and would freeze at -100 degrees. Water dash when temperature falls below 4 degrees centigrade. Water expands when temperature falls below 4 degrees centigrade. Water is most heavy at degree centigrade. Water is most heavy at 100 degrees centigrade. Ice is dash dense than liquid water. Ice is less dense than liquid water. Ice floats in liquid water due to its dash density. Ice floats in liquid water due to its less density. The breakdown of a water molecule into hydrogen ions and hydroxyl ions is called. The breakdown of a water molecule into hydrogen ions and hydroxyl ions is called ionization. For the normal functioning of dash, the presence of ions is important. For the normal functioning of enzymes, the presence of ions is important. Dash are generally dehydrated carbons. Carbohydrates are generally dehydrated carbons. Dash is present in all organic compounds. Carbon is present in all organic compounds. Carbohydrates either contain aldehydes or ketone, as dash groups attach to one of the carbon atoms. Carbohydrates either contain aldehydes or ketone as hydroxyl groups attached to one of the carbon atoms. Dash are the most abundant organic biomolecules in nature. Carbohydrates are the most abundant organic biomolecules in nature. Carbohydrate means they are hydrates of dash. Carbohydrate means they are hydrates of carbon. Dash is an example of carbohydrates. Cellulose is an example of carbohydrates. Carbohydrates are classified into dash groups. Carbohydrates are classified into 3 groups. Dash are simple sugars. Monosaccharides are simple sugars. Dash cannot be hydrolyzed. Monosaccharides cannot be hydrolyzed. Dash are easily soluble in water. Monosaccharides are easily soluble in water. Monosaccharides are dash in taste. Monosaccharides are sweet in taste. Dash have a carbon backbone. Monosaccharides have a carbon backbone. Monosaccharide having 3 carbon atoms in the backbone are called monosaccharide having 3 carbon atoms in the backbone are called trios. Monosaccharide having 5 carbon atoms in the backbone are called monosaccharide having 5 carbon atoms in the backbone are called pentose. The backbone of monosaccharide may contain dash to dash carbon atoms. The backbone of monosaccharide may contain 3 to 7 carbon atoms. Dash is the simplest monosaccharides with 3 carbon atoms. Triose is the simplest monosaccharides with 3 carbon atoms. Glyceraldehyde is triose with dash groups. Glyceraldehyde is triose with aldehyde groups. Ribose is a dash. Ribose is a pentose. The ketonic form of ribose is ribulose. Glucose, fructose and galactose are dash with same empirical formula but different structural formula. Glucose, fructose and galactose are hexose with same empirical formula but different structural formula. Molecules with same empirical formula but different structural formula are called Molecules with same empirical formula but different structural formula are called isomers. Glucose is aldehyde. Fructose is ketone. Dash are interconvertible. Isomers are interconvertible. Dash when put in water forms ring structure. Glucose when put in water forms ring structure. Dash are white crystalline powders. Monosaccharides are white crystalline powders. Dash are isomers with same structural and chemical formula but different arrangement of atoms in space. Stereoisomers are isomers with same structural and chemical formula but different arrangement of atoms in space. When two or more compounds have same structure but are mirror image of each other are called dash isomers. When two or more compounds have same structure but are mirror image of each other are called optical isomers. Dash isomers have same molecular formula but different structural formula. Structural isomers have same molecular formula but different structure formula. Carbohydrates made of 3 to 10 simple sugars are called oligosaccharides. The units or monomers of oligosaccharides are bonded together by dash bonds. The units or monomers of oligosaccharides are bonded together by glycosidic bonds. Glycosidic bonds are dash bonds. Glycosidic bonds are covalent bonds. The oligosaccharides that are hydrolyzed into simple units are called disaccharides. Dash are the most common oligosaccharides. Disaccharides are the most common oligosaccharides. Sucrose are hydrolyzed into both fructose and glucose. Dash is a disaccharide. Sucrose is a disaccharide. Dash is found in sugar cane. Sucrose is found in sugar cane. Dash is found in milk. Lactose is found in milk. Dash hydrolysis into glucose and galactose. Lactose hydrolysis into glucose and galactose. Dash is found in fruits. Fructose is found in fruits. Dash is also called table sugar. Sucrose is also called table sugar. Dash is found in our digestive tract as a result of starch digestion. Maltose is found in our digestive tract as a result of starch digestion. Maltose hydrolysis into 2 dash units. Maltose hydrolysis into 2 glucose units. Polysaccharides are dash of monosaccharide units. Polysaccharides are polymers of monosaccharide units. Polysaccharides are hydrolyzed into more than dash monomers. Polysaccharides are hydrolyzed into more than 10 monomers. The monomers of polysaccharides are glucose. Dash are tasteless. Polysaccharides are tasteless. Dash are insoluble in water. Polysaccharides are insoluble in water. Dash is a polysaccharide. Chitin is a polysaccharide. Polysaccharides act as dash for small carbohydrate units. Polysaccharides act as macromolecule for small carbohydrate units. Plants change starch into dash during photosynthesis. Plants change starch into glucose during photosynthesis. Dash is a polymer. Starch is a polymer. Dash is stored in plant cell. Starch is stored in plant cell. Dash is stored in animal cell. Glycogen is stored in animal cell. Dash is a carbohydrate found abundantly in nature. Cellulose is a carbohydrate found abundantly in nature. Dash is the stored form of glucose. Glycogen is the stored form of glucose. Dash is human body's main source of energy. Glucose is human body's main source of energy. Glycogen is a large branched dash. Glycogen is a large branched polysaccharide. Dash is the building material of plant cell wall. Cellulose is the building material of plant cell wall. Cellulose is hydrolyzed by a group of enzymes called Cellulose is hydrolyzed by a group of enzymes called cellulase. Dash is not present in human digestive system. Cellulase is not present in human digestive system. Dash fibers are example of cellulose. Cotton fibers are example of cellulose. Dash is found in the exoskeleton of crabs and insects. Chitin is found in the exoskeleton of crabs and insects. Dash is not digestible. Both chitin and cellulose are not digestible. In Dash, an amino group is attached to each glucose molecule. In chitin, an amino group is attached to each glucose molecule. Dash are used as a source of energy. Carbohydrates are used as a source of energy. During Dash, the CH bonds of carbohydrates are broken down to release energy required for cell function. During respiration, the CH bonds of carbohydrates are broken down to release energy required for cell functions. 100 ml of human blood contains dash mg of glucose. 100 ml of human blood contains 100 mg of glucose. Dash are stored in the cells as reserve food. Carbohydrates are stored in the cells as reserve food. Dash are our storage molecules to store excess amount of food and energy. Polysaccharides are our storage molecules to store excess amount of food and energy. Our bodies can digest only dash-handed sugars. Our bodies can digest only right-handed sugars. Most naturally occurring sugar molecules are dash-handed sugars. Most naturally occurring sugar molecules are right-handed sugars. Dash are most abundant organic compounds of the cell. Proteins are most abundant organic compounds of the cell. Proteins are formed of units called amino acids. Proteins are macromolecules. The carboxylic acids having amino groups are called dash acids. The carboxylic acids having amino groups are called amino acids. Dash different groups are attached to the alpha carbon. 4 different groups are attached to the alpha carbon. The central carbon present in each amino acid is called alpha. Amino groups, carboxylic groups or hydrogen are dash members present in all amino acids. Amino groups, carboxylic groups or hydrogen are constant members present in all amino acids. The dash is variable, that is either hydrogen or alkyl group present in amino acids. The R group is variable, that is either hydrogen or alkyl group present in amino acids. Due to dash, the amino acids are different from each other. Due to R group, the amino acids are different from each other. Only dash types of amino acids combine to form thousands of protein molecules. Only 20 types of amino acids combine to form thousands of protein molecules. Amino acids are dash acids having amino groups. Amino acids are carboxylic acids having amino groups. Amino acids in a protein molecule are linked by a peptide. The peptide bond links the carboxylic group to dash group of two amino acids. The peptide bond links the carboxylic group to amino group of two amino acids. When two amino acids join together, a dash is formed. When two amino acids join together, a dipeptide is formed. Peptide bond is dash in nature. Peptide bond is chemical in nature. Proteins usually have chains of dash acids. Proteins usually have chains of amino acids. The chains of amino acids in proteins are held together by dash bonds. The chains of amino acids in proteins are held together by peptide bonds. Red blood cells have a protein called hemoglobin. Hemoglobin has chains of multiple dash. Hemoglobin has chains of multiple amino acids. Hemoglobin carries oxygen. Hemoglobin has dash polypeptide chains. Hemoglobin has 4 polypeptide chains. Insulin has dash polypeptide chains. Insulin has 2 polypeptide chains. In a protein molecule, the position of amino acid specifies its function. Hemoglobin is usually dash in shape. Hemoglobin is usually globular in shape. The dash shape is due to the absence of one globular molecule in its specific position. The sickle shape is due to the absence of one globular molecule in its specific position. Red blood cells have dash shape. Red blood cells have disc shape. Normal hemoglobin molecules are flexible. A globular hemoglobin molecule has dash acid. A globular hemoglobin molecule has glutamic acid. In a sickle cell hemoglobin molecule, the glutamic acid is replaced by valine. In a normal hemoglobin molecule, there are dash chains. In a normal hemoglobin molecule, there are both alpha and beta chains. The sickle cell hemoglobin occurs due to inheritance of dash genes from parents. The sickle cell hemoglobin occurs due to inheritance of abnormal genes from parents. The sickle cell hemoglobin dash perform its function. The sickle cell hemoglobin cannot perform its function. The size of protein molecule depends on number and kinds of. The size of protein molecule depends on number and kinds of amino acids. Sickle cell RBCs are stiff. Dash protein is the most abundant protein found in vertebrate skin. Fibrous protein is the most abundant protein found in vertebrate skin. Normal proteins have dash types. Normal proteins have two types. Dash proteins form sheet-like structures in hair, nails, and skin, etc. Fibrous proteins form sheet-like structures in hair, nails, and skin, etc. The fibers of fibrous molecule have dash chains. The fibers of fibrous molecule have polypeptide chains. Fibrous proteins are dash found in hair and nails. Fibrous proteins are keratin found in hair and nails. The dash present in muscle cell are fibrous proteins. The myosin present in muscle cell are fibrous proteins. The dash of the eye is made up of fibrous proteins. The cornea of the eye is made up of fibrous proteins. Dash protein is found in the form of keratin, myosin, and collagen. Fibrous protein is found in the form of keratin, myosin, and collagen. In fibrous protein, the polypeptide chains are arranged dash. In fibrous protein, the polypeptide chains are arranged linearly. Fibrous proteins are water dash. Fibrous proteins are water insoluble. Globular proteins are water dash. Globular proteins are water soluble. Globular proteins are dash in shape. Globular proteins are spherical in shape. Dash proteins are found in the form of hemoglobin, albumin, etc. Globular proteins are found in the form of hemoglobin, albumin, etc. Albumin of egg white is formed of dash protein. Albumin of egg white is formed of globular protein. Dash proteins are present in cell membrane. Globular proteins are present in cell membrane. There are dash levels of organization of protein molecules. There are 4 levels of organization of protein molecules. The quaternary structure of protein molecule is also called hemoglobin. When amino acids are linearly arranged in a peptide chain, it forms dash structure. When amino acids are linearly arranged in a peptide chain, it forms primary structure. When a polypeptide chain of amino acids become coiled, it is called a dash structure. When a polypeptide chain of amino acids becomes coiled, it is called a secondary structure. When the coiled chains of polypeptides are arranged in 3-dimensional structure, it is called a dash structure. When the coiled chains of polypeptides are arranged in 3-dimensional structure, It is called a quaternary structure. Dash is a large-sized molecule with two or more polypeptide chains. Hemoglobin is a large-sized molecule with two or more polypeptide chains. When two or more polypeptide chains form a large molecule, it is called a dash structure. When two or more polypeptide chains form a large molecule, it is called a quaternary structure. All dash are proteins. All enzymes are proteins. In plants, proteins are stored in seeds. The important functions of living organisms are performed by protein. Dash is oxygen-carrying protein of red blood cells. Hemoglobin is oxygen-carrying protein of red blood cells. Dash present in muscle cells help in movements. Myosin present in muscle cells help in movements. Insulin is a dash. Insulin is a protein. Insulin is a dash. Insulin is a hormone. Dash speed up biological reactions inside the body. Enzymes speed up biological reactions inside the body. Without dash, food cannot be digested. Without enzymes, food cannot be digested. Lipids are dash in water. Lipids are insoluble in water. Lipids are dash. Lipids are polar. Lipids have dash oxygen as compared to carbohydrates. Lipids have more oxygen as compared to carbohydrates. Lipids are dash in organic solvent. Lipids are soluble in organic solvent. Dash is an organic solvent. Petrol is an organic solvent. Lipids are soluble in dash. Lipids are soluble in ether. The most common acyl glycerol is triglyceride. Dash is a 3-carbon compound. Glycerol is a 3-carbon compound. Glycerol is dash in water. Glycerol is soluble in water. Hydroxyl groups in glycerol are polar. Lipids have dash carbon-hydrogen bonds as compared to carbohydrates. Lipids have more carbon-hydrogen bonds as compared to carbohydrates. The acids of fatty acids react with hydroxyl of glycerol to form. The acids of fatty acids react with hydroxyl of glycerol to form triglyceride. In a cell, most of fatty acids have dash carbon atoms. In a cell, most of fatty acids have 16 to 18 carbon atoms. Fatty acids contain dash chains. Fatty acid contains hydrocarbon chains. Fatty acids may be saturated and unsaturated. Triglycerides are stored in animals as fats. Dash fatty acids have no double bond between carbon atoms. Saturated fatty acids have no double bond between carbon atoms. Palmitic acids are dash acids. Palmitic acids are fatty acids. Fats are dash at room temperature. Fats are solid at room temperature. Dash fatty acids have double bond between carbon atoms. Unsaturated fatty acids have double bond between carbon atoms. Dash acids are unsaturated fatty acids. Oleic acids are unsaturated fatty acids. Unsaturated fatty acids are dash on room temperature. Unsaturated fatty acids are liquid on room temperature. Unsaturated fatty acids are stored in plants dash. Unsaturated fatty acids are stored in plants seeds. Unsaturated fatty acids have dash 2 hydrogen per carbon atom. Unsaturated fatty acids have less than 2 hydrogen per carbon atom. Saturated molecules dash get more atom. Saturated molecules cannot get more atom. Dash have high caloric value. Triglyceride have high caloric value. Triglycerides have twice as much energy per gram as that of dash. Triglycerides have twice as much energy per gram as that of carbohydrates. Ghee is dash fatty acid. Ghee is saturated fatty acid. By replacing one dash, a triglyceride can be changed into phospholipid. By replacing one phosphate, a triglyceride can be changed into phospholipid. A molecule of phospholipid has dash parts. A molecule of phospholipid has 2 parts. The head of phospholipid is made of dash. The head of phospholipid is made of phosphate. A phospholipid has 2 dash. A phospholipid has 2 carbon tails. The 2 carbon tails of phospholipids are non-polar. Polars are dash in water. Polars are soluble in water. Phospholipids arrange themselves in double layer in the dash of cells. Phospholipids arrange themselves in double layer in the plasma membrane of cells. A long chain of fatty acids bonds with a long chain of alcohol to form dash. A long chain of fatty acids bonds with a long chain of alcohol to form wax. Wax are dash at room temperature. Wax are solid at room temperature. Wax have dash melting point. Wax have high melting point. Wax is a dash compound. Wax is a stable compound. The waterproof layer of leaves is formed by dash. The waterproof layer of leaves is formed by wax. The waterproof layer of leaves and fruits is dash. The waterproof layer of leaves and fruits is cuticle. Dash reduces the rate of water loss. Cuticle reduces the rate of water loss. Waxy layer of dash is waterproof. Waxy layer of insects is waterproof. Dash also have waxy layer on their skin. Sheep also have waxy layer on their skin. Dash are resistant to degradation. Wax are resistant to degradation. Wax are dash in water. Wax are insoluble in water. Lipids without fatty acids are steroids. Each steroid has a backbone of dash fused carbon rings. Each steroid has a backbone of 4 fused carbon rings. Each carbon ring of steroid has dash carbon atoms. Each carbon ring of steroid has 17 carbon atoms. Best example of steroid is cholesterol. Dash is a steroid hormone. Vitamin D is a steroid hormone. Dash are the steroid hormones. Cholesterol are the steroid hormones. Dash helps to regulate the sodium in blood. Aldosterone helps to regulate the sodium in blood. Terpenoids are dash in water. Terpenoids are insoluble in water. Terpenoids are soluble in dash. Terpenoids are soluble in lipids. The units of terpenoids are dash units. The units of terpenoids are isoprenoid units. Isoprenoid join by the process of dash to form different compounds. Isoprenoid join by the process of condensation to form different compounds. Dash is a terpenoid compound. Rubber is a terpenoid compound. Dash are produced by isoprenoid. Carotenoids are produced by isoprenoid. Dash are long-term energy storage molecules in plants and animals. Lipids are long-term energy storage molecules in plants and animals. Plants store lipids in seeds in the form of dash. Plants store lipids in seeds in the form of oil. Some dash are hormones which perform different functions of the body. Some lipids are hormones which perform different functions of the body. Animals store lipids in the form of dash. Animals store lipids in the form of fats. Dash is a plant hormone. Auxin is a plant hormone. Nucleic acids are dash. Nucleic acids are polymers. The units of nucleic acids are nucleotides. There are dash types of nucleic acids. There are 2 types of nucleic acids. A dash is a simple sugar. A pentose is a simple sugar. A pentose sugar has dash carbons. A pentose sugar has 5 carbons. Dash are simple sugars. Monosaccharides are simple sugars. An dash bond joins an acid to alcohol in a molecule. An ester bond joins an acid to alcohol in a molecule. In pentose sugar, the bond between phosphoric acid and hydroxyl group is dash bond. In pentose sugar, the bond between phosphoric acid and hydroxyl group is ester bond. A nucleotide has a sugar molecule attached to a phosphate group and a dash base. A nucleotide has a sugar molecule attached to a phosphate group and a nitrogen base. There are dash types of nitrogen bases. There are two types of nitrogen bases. Dash are single ring compounds. Pyrimidine are single ring compounds. Dash are double ring compounds. Purine are double ring compounds. There are dash types of pyrimidines. There are three types of pyrimidines. Thymine, cytosine and uracil are thymine, cytosine and uracil are pyrimidine. Adenine and guanine are purine. A dash is a single nucleotide. A mononucleotide is a single nucleotide. There are dash types of nucleotides. There are three types of nucleotides. The most common mononucleotide is adenosine triphosphate. Adenosine triphosphate has dash phosphates. Adenosine triphosphate has three phosphates. Adenine base linked to pentose sugar forms dash. Adenine base linked to pentose sugar forms adenosine. Adenosine bonded to single phosphate group is called monophosphate. Adenosine bonded to double phosphate group is called diphosphate. Adenosine bonded to triple phosphate group is called triphosphate. Dash is an energy-rich molecule. Adenosine triphosphate is an energy-rich molecule. Dash is also called energy currency. Adenosine triphosphate is also called energy currency. Dash provides energy to all cells of living organisms. Adenosine triphosphate provides energy to all cells of living organisms. In adenosine triphosphate, the wavy bond between phosphate group indicates dash bonds. In adenosine triphosphate, the wavy bond between phosphate group indicates high energy bonds. When adenosine triphosphate is hydrolyzed into adenosine diphosphate, it produces dash KCl energy. When adenosine triphosphate is hydrolyzed into adenosine diphosphate, it produces 7 KCl energy. In adenosine triphosphate, usually the bond between the dash and dash phosphate breaks to release energy used by the cell. In adenosine triphosphate, the bond between the second and third phosphate breaks to release energy used by the cell. Adenosine triphosphate molecules are produced during respiration. Adenosine triphosphate molecules are hydrolyzed into adenosine monophosphate. A dinucleotide has dash base. A dinucleotide has nitrogen base. The nucleobase of dinucleotide is made both of purine and pyrimidine. Adenine and guanine are called dash bases of DNA. Adenine and guanine are called purine bases of DNA. Adenine and guanine have a fused ring skeletal structure derived of Adenine and guanine have a fused ring skeletal structure derived of purine. If dinucleotide has adenine as nitrogen base, it is called dash dinucleotide. If dinucleotide has adenine as nitrogen base, it is called adenine dinucleotide. Nicotinamide is a dash called nicotinic acid. Nicotinamide is a vitamin called nicotinic acid. Adenine nucleotide combines with dash to form coenzymes. Adenine nucleotide combines with vitamin to form coenzymes. Nicotinamide adenine dinucleotide, flavine adenine dinucleotide, and nicotinamide adenine dinucleotide phosphate are nicotinamide adenine dinucleotide, flavine adenine dinucleotide, and nicotinamide adenine dinucleotide phosphate are coenzymes. Nicotinamide adenine dinucleotide, flavine adenine dinucleotide, and nicotinamide adenine dinucleotide phosphate are involved in both oxidation and hydrogen transfer. Vitamin B2 is also called flavin and riboflavin. Coenzymes exist in dash forms. Coenzymes exist in two forms. Coenzymes in oxidized form functions as dash acceptor. Coenzymes in oxidized form functions as hydrogen acceptor. Reduced and oxidized are the forms of coenzymes. Coenzymes in reduced form act as dash carriers. Coenzymes in reduced form act as electron carriers. Dash are active in electron transport chain of respiration. Both FDP and NAD are active in electron transport chain of respiration. In dash, the NAD and FAD are alternately reduced and oxidized. In respiration, the NAD and FAD are alternately reduced and oxidized. When many nucleotides are linked together, they form polynucleotide. Dash molecules are polynucleotides. Both RNA and DNA molecules are polynucleotides. DNA is made of deoxyribonucleotide. RNA is made of ribonucleotide. DNA and RNA are nucleic acids. The main function of RNA is protein synthesis. The main function of DNA is to store information. DNA molecule is a dash helix. DNA molecule is a double helix. Double helix is like a dash staircase. Double helix is like a spiral staircase. DNA molecule has a diameter of dash nanometers. DNA molecule has a diameter of 2 nanometers. The distance between the base pair of DNA is dash nanometers. The distance between the base pair of DNA is 0.34 nanometers. Pentose sugar is present in DNA in the form of dash. Pentose sugar is present in DNA in the form of deoxyribose. DNA molecule has dash strands. DNA molecule has 2 strands. Thymine in one strand is always paired with adenine. Guanine in one strand is always paired with cytosine. All four types of nucleotides are the building blocks of DNA. The backbone of DNA strand is made of sugar alternating with dash group. The backbone of DNA strand is made of sugar alternating with phosphate group. The sequence of nucleotide of one type of DNA is constant. There are dash hydrogen bonds between adenine and thymine. There are 2 hydrogen bonds between adenine and thymine. There are dash hydrogen bonds between guanine and cytosine. There are 3 hydrogen bonds between guanine and cytosine. Nearly every cell in a person's body has dash DNA. Nearly every cell in a person's body has same DNA. DNA is a dash of nucleotides. DNA is a polymer of nucleotides. RNA contains pentose sugar as ribose. RNA is formed of dash nucleotides. RNA is formed of 4 nucleotides. RNA has dash strands. RNA has 1 strand. The backbone of RNA strand is made up of phosphate and dash sugar. The backbone of RNA strand is made up of phosphate and ribose sugar. Dash is the simplest form of genetic molecule. RNA is the simplest form of genetic molecule. RNA consists of dash nitrogen bases. RNA consists of 1 nitrogen base. The bases used in RNA are adenine, cytosine, guanine, and dash. The bases used in RNA are adenine, cytosine, guanine, and uracil. There are dash types of RNA. There are 3 types of RNA. Dash RNA carries messages from DNA to the ribosome. Messenger RNA carries messages from the DNA to the ribosome. Transfer RNA transfers specific dash from cytoplasm to ribosome. Transfer RNA transfers specific amino acids from cytoplasm to ribosomes. Ribosomal RNA combines with dash to form ribosomes. Ribosomal RNA combines with proteins to form ribosomes. Chromosomes are composed of DNA and dash. Chromosomes are composed of DNA and protein. Dash is a genetic material. DNA is a genetic material. Dash is the data bank of living organisms. DNA is the data bank of living organisms. All genetic data is stored in DNA in the form of dash. All genetic data is stored in DNA in the form of genes. A dash is the unit of heredity in living organisms. A gene is the unit of heredity in living organisms. Genes are made up of DNA. A dash is a long chain of DNA having many genes. A chromosome is a long chain of DNA having many genes. Dash specify proteins and RNA chains. Genes specify proteins and RNA chains. Genetic information is transferred to next generation through dash. Genetic information is transferred to next generation through DNA. A dash is a small section of DNA. A gene is a small section of DNA. A dash is a sequence of nucleotides in DNA and RNA. A code is a sequence of nucleotides in DNA and RNA. The transfer of genetic information from DNA to RNA is called dash. The transfer of genetic information from DNA to RNA is called transcription. The transfer of genetic information from mRNA to amino acids is called The transfer of genetic information from mRNA to amino acids is called translation. When different molecules from different categories combine, they form dash molecule. When different molecules from different categories combine, they form conjugated molecule. Lipoprotein is a dash molecule. Lipoprotein is a conjugated molecule. Lipoprotein is formed of dash and proteins. Lipoprotein is formed of lipids and proteins. Nucleoproteins are formed by combination of dash with proteins. Nucleoproteins are formed by combination of nucleic acid with proteins. Eukaryotic chromosome is nucleoprotein. Dash are the basic framework of all cell membranes. Lipoprotein are the basic framework of all cell membranes. Dash are important part of brain and plasma membranes. Glycoprotein are important part of brain and plasma membranes. Dash are formed by the combination of carbohydrates and proteins. Glycoprotein are formed by combination of carbohydrates and proteins. Dash are formed by the combination of carbohydrates and lipids. Glycolipids are formed by the combination of carbohydrates and lipids. Dash are important components of plasma membrane. Glycoprotein are important components of plasma membrane. Dash are present in egg albumin. Glycoprotein are present in egg albumin. Higher plants take inorganic compounds from environment to make dash compounds. Higher plants take inorganic compounds from environment to make organic compounds. In condensation, water is removed. In hydrolysis, water is added. Common storage product in plants is dash. Common storage product in plants is starch. Common storage product in animals is dash. Common storage product in animals is glycogen. The most common polysaccharide found in plants is dash. The most common polysaccharide found in plants is cellulose. Dash are storage molecules. Carbohydrates are storage molecules. Dash has carbon, oxygen, hydrogen, and nitrogen as 4 essential elements. Protein has carbon, oxygen, hydrogen, and nitrogen as 4 essential elements. Vegetable oils and animal fats are triglycerides. Dash help in fat digestion. Lipids help in fat digestion. Triglycerides have dash fatty acids. Triglycerides have 3 fatty acids. Dash amino acids help in the formation of proteins. 20 amino acids help in the formation of proteins. Condensation is also called dehydration. Dash are atoms or small molecules. Monomers are atoms or small molecules. There are dash types of monomers. There are 4 types of monomers. The monomers of proteins are amino acids. The macromolecules of nucleotides are The macromolecules of nucleotides are nucleic acids. A bacterial cell has %% DNA. A bacterial cell has 1% DNA. Molecules having same formula but different chemical and physical properties are called molecules having same formula but different chemical and physical properties are called isomers. Dash is an enzyme used in the hydrolysis of maltose. Maltase is an enzyme used in the hydrolysis of maltose. Information is stored in DNA in the form of dash. Information is stored in DNA in the form of code. A sugar that has 5 carbon atoms and 4 oxygen atoms is called dash. A sugar that has 5 carbon atoms and 4 oxygen atoms is called deoxyribose. Dash is a sugar naturally produced by the body from food. Ribose is a sugar naturally produced by the body from food. The sugar in DNA has one hydroxyl group -RNA. The sugar in DNA has one hydroxyl group less than RNA. Starch is a polysaccharide. Starch is a storage polysaccharide. Peptide bonds are Peptide bonds are rigid. The shape of human red blood cell is called dash. The shape of human red blood cell is called biconcave. Deoxyribose attached to a nitrogenous base is called dash. Deoxyribose attached to a nitrogenous base is called nucleoside. A nucleoside attached to a phosphate group is called nucleotide. A molecule of simple sugar having 5 carbon atoms is called pentose. The nitrogen-containing bases of nearly all nucleotides are derivatives of heterocyclic compounds. The nitrogen-containing bases of nearly all nucleotides are derivatives of 3 heterocyclic compounds. The heterocyclic compounds which form nitrogen bases of nearly all nucleotides are the heterocyclic compounds which form nitrogen bases of nearly all nucleotides are pyrimidine, purine, and pyridine. Guanine is similar in structure to adenine. Dash is used in RNA in place of thymine. Uracil is used in RNA in place of thymine. Pentose are monosaccharides. Dash is the most basic form of carbohydrates. Monosaccharides is the most basic form of carbohydrates. Dash cannot be broken down by hydrolysis. Monosaccharides cannot be broken down by hydrolysis. Dash are used to produce and store energy. Monosaccharides are used to produce and store energy.